We've talked about the structure of DNA and how DNA is made up of the monomer called nucleotides, and nucleotides are made up of a phosphate and sugar and a base each, where the bases connect with one another to join the two strands of DNA. Adenine pairs with thymine and cytosine pairs with guanine. Now we're going to take a look at how DNA copies, therefore DNA replication. So DNA replication is a process by which DNA is copied to make more DNA. And we talked a little bit about DNA needing to copy to make more when we talked about cell division. So a process will occur in the nucleus, which makes sense because the DNA is found in the nucleus. We're going to end up with double the DNA, so more DNA than we started with, and we end up with an exact copy. So now we have two sets of DNA that are exact copies of each other. We said earlier that DNA is copied to make more, so there's enough for new cells when new cells replicate during mitosis and meiosis, and that DNA is copied in interphase during the S phase. Now we'll have enough DNA to make both pieces of our chromosome so they can be distributed into the new cells, and the new cells that are made will have all of the pieces of DNA that are needed. So there's a few steps to DNA replication. Our book talks about 4 of them, so we'll stick with 4. Sometimes they are grouped differently and maybe only listed as 3, for example, but it's the same basic concept of what is happening. So in the first step, DNA actually has to be unzipped. That means we are taking the DNA molecule apart so we can access the bases that are in the middle. So imagine that our DNA ladder has to be opened and pulled apart between the bases so we can read the information. It's kind of like opening a book in order to read the information in the book. You can't read the book when it is closed. We can't read the DNA when it is closed. So in this picture here, DNA is being unzipped. Now there's an enzyme that helps to do this unzipping, and it's called helicase. So helicase is the name of the enzyme, and it binds to the DNA. It is not pictured here, and it will break the hydrogen bonds between the phosphate groups, one right after the other. So in this picture, it'd be going from bottom to top, breaking the bonds between these bases as it unzips the molecule, and you see the molecule coming apart from one end to the other. Each of these strands, now showing in this case exposed bases at the bottom here, will act as a template to build a new strand. So both strands will be read and new strands will be made in order to complete the DNA. In step 2, another enzyme called DNA polymerase is going to read the DNA molecule, and then it can start to bond new nucleotides. The nucleotides that are attaching are pairing up with their matching bases. So if we have an A exposed, a nucleotide carrying a T or thymine will bond. And these nucleotides are called free nucleotides because they are not already attached, and they are going to bond to their complementary base. So here is our unzipped molecule. The polymerase would bind to both sides. There'll be 2 polymerase molecules, and you can see these free nucleotides are bonding by matching up complementary bases. So here is an adenine, a nucleotide with a thymine bonded. Here is a cytosine, a nucleotide with a guanine is about to bind. That will happen on both sides, matching up complementary base pairs in order to complete the DNA molecule. The bonding is happening between the bases, so we're forming hydrogen bonds between those bases. Finally, our sugar backbone needs to bond, so that means the sugars and the phosphates in the backbone of our DNA molecule connecting one nucleotide to another to kind of solidify that new molecule that's being formed. DNA polymerase is the enzyme that will also do this step. So our helicase unzip the molecule by breaking the bonds, and our DNA polymerase will read, base pair, and then solidify those bonds, connecting one nucleotide to the next. So these are strong covalent bonds between sugars and phosphates of nucleotides that are next to each other. So this will kind of complete those strands and make them nice and solid and strong. And now we're going to have 2 complete DNA molecules. Each DNA molecule will consist of an old strand, consider the parent strand, an original strand, and a newly built daughter strand. So we're going to have 2 pieces. This process is considered semi-conservative because we are only building one strand and we are conserving the original strand. And there is a figure in our textbook that might help illustrate this. I'll show you one here as well. So this image kind of steps through in just 3 pieces. So to the left, we have our first DNA molecule. This light purple structure is starting to unzip that molecule. This is that helicase that's breaking the bonds between the bases from the bottom to the top in this picture. The second picture shows that helicase is moving along, but then this light blue represents the DNA polymerase. Notice there's a DNA polymerase on each of the exposed strands. It's reading the bases that are there, base pairing new free nucleotides to help build that new strand. And it will finally connect the nucleotides to one another by bonding those sugars and phosphates of nucleotides next to each other to make that solid molecule. And then at the end here, we end up with two separate strands of DNA molecules, two separate molecules of DNA that are the same as one another because we read the original bases and matched them up. So we can see that these two molecules will be the same as each other. And each molecule would consist of a new piece that was just built and an old piece from the original DNA molecule. Remember, the process is semi-conservative. So let's take a look at how the two molecules will compare to each other. So this would represent our original DNA molecule, where all the pieces are going to be dark blue in the picture. Our helicase unzips the molecule, so it breaks the bonds, those weak hydrogen bonds between the bases. And then the polymerase will bind. It will read the bases that are there. So in this case, it's going to read the A on this side and the T on this side. And then it will bond those free nucleotides. So we have an A, we're going to match a T. We have a C, we're going to match a G. On the other side, we have another DNA polymerase. There's a T, we're going to match an A G, we're going to match a C. Notice we're building both sides. We also notice that the sugars and phosphates of that new strand are not connected yet. And finally, our polymerase, the DNA polymerase, will now solidify those sugars and phosphates. So they will connect the sugars and phosphates of nucleotides next to each other to make the strand solid. And we end up with two molecules of DNA. So here's one on the left and here's one on the right, each one with an old strand or parent strand that is in dark blue, here and here, and a new strand that is just built, a daughter strand that is in light blue here and here. Notice that our DNA strands or our DNA molecules are identical to one another. So let's take a look and see if we can practice. So if a DNA molecule was to be replicated, each strand of that DNA molecule, once it is unzipped, will be red, and base pairs will be connected in order to make that new complementary strand. So if this was our original DNA strand, AAC, GGC, TAC, AGC, ATA, what would be the complementary strand that is built? Take a moment to pause the video and see if you can figure out the complementary strand. The first one's been done for you. So if there was an A on the one side, the polymerase would add a thymine, a nucleotide carrying a thymine on the other side. Pause the video and see if you can make your prediction. All right, let's see how you did. So our complement nucleotides would have the complementary bases to what we have. So you'll see that if we have an AAC as the first group of three, the first group of three on the next side, on the complementary side, would be TTG, and so on. All right, so here is an image showing you kind of a snapshot in the middle of DNA replication. You do have a few things to try to label. Let's see if we can go over what they are. So here we have a chromosome, and our chromosome, if we unwind that and take a look more detailed, more in depth, we can see how we have our double helix here. Notice this strand right here will be a template in order for us to build a new strand of DNA, as well as this strand here will be another template. This right here represents our helicase molecule that's unzipping the molecule by breaking the bonds. Notice these bonds have already been broken, and these have not been broken yet. This orange-yellow blob here, there's one on this strand here, there's one on the bottom strand as well. That is our DNA polymerase. Our DNA polymerase is reading our strand that is exposed, base pairing our nucleotides, and then solidifying our backbone as well. It has lots of jobs. Notice we have our free nucleotides. This represents the base, okay, and this represents the sugar and the phosphate. Notice that one strand is following our following our helicase, and the other strand is actually moving in the opposite direction. There's a video you're going to watch about the leading strand and the lagging strand. DNA, you might remember, can only be read in one direction. We talked about that a little while ago. So you'll see that one strand is actually built faster than the other strand. But as we continue, we can see we're going to end up with this fully formed strand of DNA, and this will eventually be an identical fully formed strand of DNA as well.